Welcome to Summarize Science. Have you ever wondered how scientists look at the unbelievably small world of cells and atoms? Traditional microscopes face a tough choice: get a super detailed image or keep the sample alive. Getting high resolution often means blasting the sample with high-energy electrons, which can damage or destroy delicate things like living cells. But what if you could have both? Researchers have just introduced a groundbreaking new technique called Optical Near-field Electron Microscopy, or ONEM. Here's a simplified look at how it works. Instead of hitting the sample with damaging electrons, they shine a gentle beam of visible light on it. This light creates an intricate, high-resolution light pattern right behind the sample. This pattern then hits a special, ultra-thin sheet called a photocathode. The photocathode acts like a converter, turning the light pattern into an identical electron pattern. An electron microscope then safely images these new electrons, creating an incredibly sharp picture of the original sample, which itself was never touched by electrons. The result is a resolution of 31 nanometers, which is 13 times smaller than the wavelength of light they used. So, what can you do with this new superpower? First, they looked at tiny, 100-nanometer silver cubes. Depending on how they polarized the light, they could see these unique and beautiful plasmon patterns light up, something you can't see with a normal optical microscope. They could even tell the difference between a nanocube and a nanosphere just by these patterns. But the most exciting part is what they did next. They created a tiny, sealed liquid cell to look at living things in their natural environment. In this amazing footage, you are seeing a live coli bacterium swimming around in a nutrient broth. The researchers were able to track its path and even determine its 3D orientation as it tumbled through the liquid. And because the technique is so gentle, the bacteria were still alive and swimming hours later, completely unharmed. Finally, they used ONEM to watch a chemical reaction in real time. Here, you can see tiny copper crystals growing on a gold surface as an electrical potential is applied. They captured the birth and growth of these nanoclusters, frame by frame. So let's recap the breakthroughs here. ONEM provides damage-free imaging with a resolution far beyond what light alone can achieve. It works in both vacuum and liquid, allowing scientists to see everything from nanomaterials to living cells. And it's fast enough to capture these processes as they happen, creating real-time movies of the nanoworld. The future implications are enormous. Imagine watching proteins work inside a cell, seeing how medicines interact with viruses, or developing more efficient batteries by watching the chemistry unfold. This technology opens the door to seeing the unseen and understanding the fundamental processes of life and materials science in a whole new way. Thanks for watching Summarize Science.